Hello student, myself is Dr. S.V.K. Khairnar, Head and Associate Professor, Department of Botany, MEP Samaj, S.V.K.T College, Deolali Camp. Student, in the series of video, we were studying the different topics of botany. So today also we will go into studying a next topic of our botany. Today's lecture is for a SYBSc student from the paper Plant Anatomy and Embryology. So in the today's lecture we will going to learn or we will going to study the Megasporangium and mega types of ovule. Before that we will just remind what we were study earlier, that is in the form of recap. So earlier the Megasporangium, we were study the microsporangium and development of male gametophyte. In the angiospermic plant, the flower possesses the male reproductive structure and female reproductive structure. The male reproductive structure produces the microsporangium and male gametophyte, whereas the female reproductive structure produces the megasporangium and female gametophyte. So in the today's video we will going to see about the megasporangium, that is what the reproductive structure with concern to female reproduction or female reproductive organ. So our today's session agenda is, today we will going to see the structure of ovule in the angiospermic plant, and also on the basis of position of micropyle and chalazal end or stalk, how many kinds of ovules are there, or how many kinds of ovules are found in the angiospermic plants, that we will going to learn today. So what are the learning objectives of our today's video? The learning objectives of our today's video are, the angiosperm is a very vast plant group and today it is a dominant on the earth. So about their reproduction or about their embryology, we must have to get understand what is the structure of their ovule, and that's why one of the objective of our today's video is to know about the structure of ovule in the angiospermic plant. And also the next objective of our today's session, that is we have to get understand or we have to get study the different kinds of ovule found or occurs or develop in the angiosperm plants. So introduction to Megasporangium. I have already told in the previous video that the microsporangium and megasporangium are the reproductive structures of angiospermic plant. They may develop in a same flower or they may develop in the different flowers. So microsporangium that possesses the male reproductive structure, whereas the megasporangium that possesses the female reproductive structure. In angiospermic plant or in their flower, the female reproductive organ is a innermost or last whorl, and that is identified or recognized as a carpel or pistil. It is also called as a megasporophyll, and this carpel or pistil mostly contains a tip, so rounded sticky tip, or that is a receiving center for the pollen grain called as a stigma, and this stigma is joined with the basal swollen portion, that is a ovule, with the help of long cylindrical elongated structure, and that long cylindrical elongated structure is called as a style, and at the base there is a swollen basal portion that is called as a ori. These are the three typical parts found in a female reproductive organ, that is a carpel or pistil. Ori is a basal region of carpel that contains one or more ovule, and that ovules may arranged on the placenta by means of a different kinds of placentation. There may be a basal placentation, central placentation, axial placentation, precentral placentation, etc. So each ovule has a embryo sac. Inside each ovule there is a embryo sac, and that embryo sac develops from the functional megaspore within the nuclear tissue of the ovule. The functional megaspore is developed inside the ovule, inside the nuclear tissue, and there hence the ovule is referred as a megasporangium. That is what a female reproductive organ or female reproductive structure. So first of all, we will understand the in general what is a structure of ovule. What are the different parts of angiospermic ovule, that we will get understood. You can see the image. In the image, the structure of typical angiospermic ovule is drawn, and that are showing the different parts that are labeled. So ovule is a oval shaped multicellular body with stalk. Ovule is nothing but a multicellular structure, somewhat oval in appearance, and it is having a multicellular stalk. The basal portion of ovule is identified or recognized as a chalaza or chalazal end, whereas the upper portion that is identified as a microcoil. So normally in the Anaptropus ovule, the microcoil is upward facing and chalazal end is downward at the base. But in the image, this is what the inverted kind of ovule, and that is why you are finding here the chalazal end is goes upward and microcoil is come downward. But you can see the typical ovule has a microcoil, that is what the way for entering the pollen grain or dusting the pollen grain on the mouth of the embryo sac. Then inside there is a nucellus tissue. These are the nutritive tissue. The entire ovule is get covered by the outer and inner integument. That is what a protective covering, and these outer and inner integument, where they join or where they come near, that particular portion is identified or is called as a micropylar end, and there where there is a small way is developed for the entry of the pollen grain. Also you can see there is a multicellular stalk of ovule, that may also called as a funiculus. The chalazal end is what, that is a lower portion near to funiculus or stalk in the other kinds of ovule. Inside the nucellus there is a presence of embryo sac. This embryo sac may be again of different type on the basis of how many nucleus are present inside the embryo sac. So in general the embryo sac may be 8 nucleated. From that 8 nuclei, at the center there are secondary nucleus. Towards the chalazal end there are antipodal cells, and towards the micropylar end there is a egg cell and synergids are there. So this is what about the typical angiospermic ovule structure. Now after studying this ovule structure, I have already mentioned you that there are about 5 kinds of ovules are found in the angiospermic plant, and these categories or types of the ovules are prepared on the basis of position of micropylar end, funicle or stalk. How the micropylar end and funicle are present in a particular ovule, and accordingly that ovule has been given a particular type name. So in general there are 5 kinds of ovules are found in the angiospermic plants. So on the basis of direction of the micropylar region and chalazal region of a mature ovule, these types are made. So first type of ovule that is what called as a Arthropus ovule. So you can see the image is giving or highlight a picture of Arthropus ovule. In the Arthropus ovule, the micropylar end and chalazal end and funicle, all these three parts are lie in a one line, and this is what called as a Arthropus ovule. And this kind of ovule is occurs in the plants of family Polygonaceae and Piperaceae. You can see the image of Arthropus ovule. The micropylar end, that is what the ovule is in the vertical position, or it is a upright. The micropylar end is at the upper direction. Also the funicle and chalaza is also in the same line, and these are the characters of identifying this Arthropus kind of ovule. Here also you can see the ovule having a integuments, then embryo sac, nucellus, chalazal end and funicle or stalk. So this is what a first kind of ovule, and it is named as a Arthropus ovule. The next type of ovule, that is what considered as a Anatropus ovule. What happened? The Arthropus ovule is just get inverted. So this is what inverted ovule having a bent funicle. Its funicle is get bent down, and that's why what happens, the micropylar end is directed to the downward direction, and chalazal end is also directed to the upward direction. So that's why the chalazal end is go up and micropylar end is come down. This is because of a bent funicles. And this kind of ovule is a common type in the plants of monocots and dicotyledons. So you can see here the micropylar end is at the downward position, chalazal end is at the upward position, and this kind of ovule is identified as a Anatropus ovule. The third kind of ovule, that is called as a Amphitropus ovule. So here again the ovule is somewhat get turn in the different direction. Ovule is in the horizontal position. So it is what right angle to the stalk or funicles. The ovule is what right angle to stalk or funicles, and this kind of ovule is identified as a Amphitropus ovule. Here micropyle and chalaza are lateral in position. You can see the micropyle end is at the one side and chalaza is at the another side. So they are lateral of one another, and this kind of ovule is identified as a Amphitropus ovule, and this kind of ovule is found in the aquatic plants like a lemna. You can see the picture of Amphitropus ovule showing the micropylar end, chalazal end, then integument, nucellus, embryo sac, and funiculus, funicle or stalk. Now we will go to the next type of ovule, and this next type of ovule is called as a Campylotropus ovule. So this Campylotropus ovule is again somewhat direction is get changed. So it is a curved embryo. Inside this ovule, the embryo is curved, and that embryo placed transversely in the ovary, and ovule is also bent like a horseshoe. The ovule is becomes a horseshoe shape. So the micropylar end and chalazal end, because of bending, they come close to one another, and this kind of ovule is identified as a Campylotropus ovule. You may get this ovule in the plant of family Campylotropus like a Campylotropus. So in the figure you can see the structure of Campylotropus ovule, where the embryo is placed transversely, and also the ovule is get bent down like a horseshoe shoe, and that's why the chalazal end and micropylar end, they come close to one another. The next type of ovule, that is called as a Cersino Tropos ovule. Again here the position of ovule is somewhat changed. Here the ovule is with a coiled body due to rapid growth of ovule at one side. At initial stage the ovule is in a straight position, then it turns down, and again it turns upward direction. So the micropyle get turn upward again. Then example is what plumbago or opuntia. So this is what a Cersino Tropos kind of ovule. Summary. So now we in this video we study the Megasporangium, general structure of ovule and kinds of ovule. So we will just summarize what we learn. So here each ovule has a embryo sac develops from functional megaspore within the nuclear tissue of the ovule, hence the ovule is referred as a Megasporangium. Ovule is a oval shaped multicellular body with a stalk. Basal portion of ovule is called as a chalaza. Upper end is called as a micropyle. On the basis of direction of micropylar region and chalazal region of the mature ovule, ovules are categorized into 5 types, and that 5 types are Orthotropous ovule, Anatropous ovule, Amphitropus Rule, Campylotropus Rule, and Cersino Tropos Rule. So this is what a summary of our today's topic. So what are the expected assignment on this particular topic? So there may be a assignment like draw a label diagram of typical angiosperm ovule. So you have to draw the diagram of typical angiosperm ovule and you have to label the different parts. The second assignment will be like a describe different types of ovules observed in angiosperm. So here you have to answer. So in angiosperm, on the basis of direction of micropyle and chalaza, 5 types of ovules are there that you have to mention, and you have to describe each kind of ovule. So this is what our today's topic, Megasporangium, and under that we learn the typical ovule and types of ovule. What we will going to learn in the coming video. So in the coming video again we will going to learn what is mean by megasporogenesis and what are the different types of megaspore tetrads. Okay, thank you.